Hello, my name is Nicholas Craddock, and I am a graduate assistant under Dr. Jennifer Bouldin, and my research project is the pasture use effect on water quality of subwatersheds of the Buffalo National River in Arkansas. Just a little bit of background, the Buffalo National River was designated as an extraordinary resource waterway by the Arkansas Department of Environmental Quality in 2008, and it earned this designation due to its primary and secondary contact activities and because it is an economic resource to local municipalities. Of the subwatersheds of the study, 51 to 68% of the land surrounding them is forested, while 16 to 28% of the remaining surrounding land is used as pastures. It is a known fact that runoff from pasture land has both direct and indirect negative impacts on water quality parameters, such as pH, dissolved oxygen, nutrient loading, fecal coliforms, etc. Objectives of this study are to measure water quality parameters of each subwatershed and identify differences in water quality at the headwater and outlet site of each subwatershed, and in the end to determine if subwatersheds meet Ozark Highland State water quality standards. To do this, we took once weekly samples at the headwater and outlet site of 4 different subwatersheds, these sites being Bears Outlet Creek, Brush Creek, Calf Creek, and Tomahawk Creek. To get this data, we would use a bucket to collect water, and then in the field we would measure parameters such as pH, temperature, conductivity, and dissolved oxygen, and flow. And then when we got back to the lab, we would analyze the data and the sample for turbidity and total suspended solids. Through the analysis of data collected, we found that for the parameters pH and conductivity, every subwatershed has a significant difference between the upstream and the downstream sites, except for Tomahawk Creek. Though for conductivity, the only site that had a significant increase between the upstream and downstream site was Calf Creek, with an increase in conductivity between headwater and outlet, while Bear Creek and Brush Creek both had a decrease in conductivity between the upstream and downstream sites. Analysis of turbidity showed that Bear Creek and Brush Creek were the only subwatersheds that had a significant difference between the upstream and downstream sites, even though the turbidity decreased on a downstream gradient for both of those sites, which we are currently trying to come up with a reason why that may have happened. But at the same time, Brush Creek was the only subwatershed with a significant difference in total suspended solids between the upstream and downstream sites, and as in the case of turbidity and conductivity, there was an actual decrease between the upstream and downstream site. This is an ongoing project, and we are still analyzing nutrient data and counting fecal coliform data, and will continue to do so for the duration of this project. So more data may become available and may change our overall outcomes, so we will see what happens there. But as of right now, I would like to thank my partners in this project, which are Dr. Jennifer Bouldin and Jesse Brown, along with our funding for this project, which comes from the United States EPA and the Arkansas Natural Resource Commission, and as well as the National Park Service for their assistance with locating the sites and keeping the roads clean and accessible so that we can continue to get to our sites and collect data. So, thank you for listening.